Next up, we've got Kasia Ellsworth, who's an associate lab director at Rady Children's Institute for Genomic Medicine. She's completed fellowships in both clinical biochemical genetics and clinical molecular genetics at the Greenwood Genetic Center in Greenwood, South Carolina, and is certified by the American Board of Medical Genetics and Genomics in both specialties. She earned her PhD in molecular pharmacology and experimental therapeutics at Mayo Clinic College of Medicine in Rochester, Minnesota. Thank you, Kasia.
Thank you so much for the introduction. So today I would like to focus on how us, Rady Children's Institute for Genomic Medicine, utilizing tools such as Mastermind in automating our variant curation in the very unique setting in Pediatric ICU. So Rady Children's Institute for Genomic Medicine is actually embedded within Rady Children's Hospital, which is the largest hospital in California. We began our genome sequencing in July of 2016 and are currently a CLIA certified and CAP accredited laboratory. You can see that our mission is to enable the prevention, diagnosis, and treatment of childhood diseases through genomic and systems medicine research, with the goal to make genetic testing fast, easy, and routine care for diagnosing and delivering precision medical care to critically ill babies and children. So over here is illustration of, sorry, I went to the next slide. Our focus is on the intensive care unit in the pediatric setting, as I just mentioned, where many of the sick children end up. This setting provides rather a very unique opportunity to implement precision medicine and to have significant impact on the lives of children. Okay, you can see the pointer. So you can see over here, roughly about 10% of children end up being with severe chronic illness. However, that number accounts actually for about 70% of the hospital costs. Thus, to understand this population of patients is crucial to not only benefit the patients themselves, but also for the hospital economics. So over here is illustrated our current standard of care. You can see that roughly about 44% of children or neonates born would end up in our community hospitals that end up in the NICUs or PICUs, so they require a critical care. And while they're admitted within this intensive care unit, interim empirical treatment as well as search for etiological diagnosis will start, and those babies can then improve or worse, but meantime, they can get better and be discharged home, or there will be a palliative care decision made, and unfortunately, some of those children end up dying. However, if we are able to improve their condition, and modify the treatment, the outcome can be very different. So this is how we envision the future of the ICU care units, where critically ill children are admitted to the hospital. And while the search for etiological diagnosis and empirical treatment are underway, about 40% of those children, of the 4% that are admitted to the intensive care units, would be eligible for rapid whole genome sequencing. And based on the data that we gathered so far, we expect that about 20 to 50% of those kids will end up having actually a genetic diagnosis, followed by a higher percentage of those children where diagnosis will inform precision treatment, leading to avoidance of mortality and long-term morbidity and shorter clinical stay with decreased hospital costs. And our median turnaround time to deliver the genetic diagnoses from sample accession to preliminary diagnosis is around about 3 days. So here's the overview of our process that we perform today at our institution. We collaborate with more than 20 children's hospitals nationwide, where consent and enrollment takes place. At this time, we also receive patient phenotypic information, and Once the sample received and DNA is isolated, sequencing takes place. We perform sequencing on Illumina's NovaSeq, and then alignment and variant callings being made for SNVs and indels via Dragon, and for the copy number variants via DNA Nexus and our in-house developed pipeline. Then the combined VCF files are annotated in Fabric, where filtering and interpretation and reporting takes place. And once the report is generated in Fabric, is being uploaded into the electronic medical records. This is a clinically validated rapid whole genome sequencing workflow. And if any of the variants require confirmatory testing, we perform that in-house as well. And again, our median turnaround time is about 3 days from the sample accession to the generation of preliminary results that is being uploaded into medical records. So now I want to focus on the case analyses. You can see that there are 3 major hubs that are part of the case analyses, including the variant prioritization, where ranking of the variants takes place in our interpretation software that we are using. Next, variant curation, where we research variants for evidence of pathogenicity. And then if we decide to report any of the variants that we identified, once they're curated and all the evidence is gathered, then we classify them according to HGMD criteria. But this is really area where we spend a lot of time on, and truly, by improving and automating some of the variant curation parts, we are able or contribute to decrease in the turnaround time. And that's really where Mastermind comes in place. You can see, as Dan mentioned in his talk, that there are various databases and tools that we use for the variant curations. And one of them now incorporated in our workflow is Mastermind. So to illustrate that, I would like to share with you this one case where truly, being able to automate the process and decrease the turnaround time to deliver the final results made a huge difference. So this is an ultra-rapid case where fast turnaround time really does matter. That was an 8-day-old baby boy who presented due to a newborn screen being positive for SCID and was admitted to the NICU. That was the DUO analysis, as the baby was born from egg donor via IVF. And meantime, when we received the sample into our laboratory, the flow cytometry confirmed in this baby sample absence of T and B cells. So therefore, his SCID was classified as T negative, B negative, and NK positive SCID. So this is essentially a screenshot of our analysis software. And you can see when we started the analysis, we input what the HPO term is for the main phenotype associated with patient's presentation. And then while we start curating the variant, this is a particular snapshot of looking at the copy number variants. We identified this large deletion encompassing 3 exons within the gene called DCLRE1C. So then we're curious, what is this gene all about? When we look into ALMEM, then we could see that this gene is associated with autosomal recessive SCID, and an estimated 4% of severe combined immunodeficiencies is caused by pathogenic changes in this particular gene. And based on what we gathered so far, based on the patient's phenotype, the match was really, really strong. In addition to having absence of B and T cells, this baby also had chest X-ray showing absence of thymus, which was really consistent with this disease. Infants with this disease usually present a little bit later in life, with disease presentation being due to severe recurrent viral, bacterial, or fungal infections and failure to thrive. Our baby was doing really well in the NICU, so we got him, I guess, prior to severe presentation. When we look further in some of the databases, we could see that the deletion that we identified was previously reported in affected individuals. Therefore, the classification for that was pathogenic. However, as I mentioned, this is a SCID that is autosomal recessive, we continue analysis of the genome, and we identified another hit in this gene. That was a missense variant. Therefore, we could see that it was not previously reported. It was absent from the population, and in this case, because it was dual analysis, was inherited from the father. The deletion was not. So next step, when we do perform the case analysis and variant curation, we open different sources that we have available. And in silico analysis, in this case, for this particular missense trace was pretty strong. Then we look at the analytic validity of this call, and you can see, it's a little bit of a skewed call in this case. And this is actually due to the fact that this particular gene has a pseudogene that is homologous to the regions of exon 4 and 6 to 9, and this particular variant is within exon 6. Therefore, the skewing is most likely due to the pseudogene presence. Most importantly, what we actually learned from our immunologists was that this gene is notoriously difficult to sequence, and variants could potentially be missed or filtered out. But we got it. So then we look into first HGMD to see whether or not it was previously reported, and it was absent. So then we continue the curation of the variant. And you can see that actually we have in our variant curation workflow, as well as in our software that we analyze the cases, this particular link out with the letter M, and essentially this is a link out to the Mastermind. So this variant is present actually in the Mastermind, means that it has some publications associated with it. So this window pops up when you click on it, and you can see that indeed, there's one publication that talks about this particular amino acid position. When we open the article, we could quickly access and read the data, and it showed that in vivo and in vitro studies were performed. This variant's name was actually under the protein's name, which is Artemis, rather than the full gene name, which is different. What we learned from reading this paper is that this particular amino acid position is actually one of the amino acid positions that are essential for the protein catalytic activities, and therefore we were able to evoke evidence for functional evidence. So both variants then, when confirmed via orthogonal testing and reported as likely pathogenic and pathogenic, we could not obviously provide the phasing of this variant, as we did not have maternal sample available. However, one of them was detected in the dad, and the other was not. So based on this clinical presentation of the patient and molecular findings, the clinical team actually agreed that this is indeed a patient's diagnosis, and very likely it was done prior to onset of severe symptoms. Moreover, this particular type of SCID is difficult to transplant by conventional BMT, and because the baby was diagnosed quickly and prior to onset of symptoms, he was actually eligible to be enrolled in the gene therapy trial, and so far is doing great post-transplant of the genetically corrected cells. And this is just an email we received from the clinician taking care of the patient, saying that knowing the genetics rapidly made this gene therapy available and able to be performed prior to any infections. So that was really, stunning, I guess, results, and the strength of the Mastermind of providing us, additional information for variant classification in a timely manner. This table summarizes some of the studies that we, performed in other cases. This is a rather large table, but you can see that, for several cases and a subset of variants that we looked at, Mastermind references were the number of references from the Mastermind were, very high as opposed to HGMD references. Moreover, subset of those references were very useful for our curation. And what is very important that, for many of these particular variants, that was very important as those were diagnostic variants that were reported on the patient's reports. So in summary, I show that there are there's clinical utility of performing rapid whole genome sequencing tests in the pediatric ICU, specifically that rWGS can help to guide medical management of genetic disease. And moreover, the automation of the variant interpretation workflow decreases turnaround times, specifically, by patient phenotyping as well as literature curation. And with that, I want to say that we hope that the most acutely ill newborns and children have access to this test as routine standard of care, as that makes a difference. Thank you to the RCIGM team, as well as to all our collaborators to make it happen. Thank you.